Hi guys, I'm on vaccines.gov website, and so if you're trying to understand what are different types of vaccines, and it might be a little bit hard in the beginning, but I'm gonna give you a very short lesson, and it'll stick to your brain for a long period of time. So there are different types of vaccines, but we're gonna focus on four main types of vaccine: live attenuated, inactivated, and we're gonna focus on three actually, and the recombinant type. So as you can see, the live attenuated vaccine, it's a weakened form of the germ. Um, so it is a live vaccine, and they are pretty similar to the natural infection. Uh, they create a very strong, long-lasting immune response, and just one or two doses of most uh, live vaccines can give you a lifetime of protection against a germ and the disease. So just one or two doses, but it also has some limitations. It does contain a small amount of weakened live virus, so some people should talk to their healthcare provider before receiving them. People with weakened immune systems, long-term health problem, and who have had an organ transplant. So these are the people they cannot take or should consider talking to their healthcare provider. They need to be kept cool so they do not travel well. That means they can be used in countries with limited access to refrigerator. So they protect against rotavirus, smallpox, chickenpox, and yellow fever. So one or two doses would be enough for, and it'll give you a lifetime, a long-lasting, strong and long-lasting immune response. And it can give you a lifetime of protection against a germ and the disease. So inactivated vaccine uh, use the killed version of the germ that causes a disease. And so the inactivated virus that doesn't provide as strong as live immunity, as strong as live vaccine, and you may need several doses over time to like booster shots to get ongoing immunity. So I think for coronavirus, this one is good enough. The inactivated vaccine, you just get like a shot every season, and and it'll give you the protection against hepatitis A, flu, polio, and rabies. And the third category is recombinant, and this is very interesting. This the third category. Um, so what it does, it uses specific pieces of the germ, like its protein, sugar, or a capsid. And because these vaccines use only specific pieces of the germ, they give a very strong immune response that's targeted to key parts of the germ. They can also be used on almost everyone who needs them, including people with weakened immune systems and long-term health problems. So it can be used on almost everyone who needs them, people with weakened immune system and long-term health problem. Um and there are only specific pieces of the germ, and they can give you a very strong immune response that's targeted. So this category would be very suitable. Recombinant category would be very suitable for um, coronavirus. And the same thing here, you need booster shots to get ongoing protection. And the and the and so it is used for influenza type B, hepatitis B, whooping cough. I don't shingles. I don't know what these are, but anyway. And toxoid vaccine use a toxin harmful product made by the germ that causes a disease. They create immunity to the parts of the germ that causes a disease instead of the germ. And the future of vaccines. DNA vaccines are easy and inexpensive to make, and they produce strong long term immunity. Nucleic acid vaccines. Another investigational approach to vaccination involves introducing genetic material encoding the antigen or antigens against which an immune response is sought. A DNA plasmid vaccine comprises a small circular piece of DNA called a plasmid that carries genes encoding protein from the pathogen of interest. The manufacturing process for DNA plasmid vaccine is well established, allowing experimental vaccines to be quickly developed to address emerging or re-emerging infectious diseases. Um, NIAID Vaccine Research Center has developed candidate DNA vaccines to address several viral disease threats during outbreaks, including SARS coronavirus in 2003, H5N1, H1N1 pandemic, influenza, and Zika virus. The time from selection of the viral genes to be included in the vaccine to initiation of clinical studies in humans was shortened from 20 months with SARS-CoV to slightly longer than 3 months with Zika virus. Vaccine based on messenger RNA and intermediary between DNA and protein also are being developed. Recent technological advances have largely overcome issues with the instability of mRNA and the difficulty of delivering it into cells. Some mRNA vaccines have demonstrated encouraging early results. For example, NIAID supported researchers develop an experimental mRNA vaccine that protected mice and monkeys against Zika virus infection after a single dose.